Hello, this is Tanawong Maison, PhD student in Dr. Han's Stein Lab. My research presentation today is amino acid digestibility in canola meal, 00 rapeseed meal, and 00 rapeseed expellers fed to growing pigs. Here's the outline. Today we have brief introduction talking about background of canola and rapeseed, and I will also talk about factors that could affect the quality and nutritional values of canola and rapeseed meal. Then I will have materials and methods, results, and conclusion for this experiment. Canola and rapeseed are an oilseed in genus Brassica. Actually, rapeseed has been developed by plant breeder to obtain low erucic acid in oil and low glucosinolates in meal. Then they changed the name from rapeseed to canola, especially in North America, and they may call 00 or 00 rapeseed in Europe. As you can see here in this picture, on top left, this picture shows how canola and rapeseed growing field look like. And on the top right, you can see canola and rapeseed are in round shape with different color of seed coat. It could be black, brown, red, or yellow. After oil extraction, they will have canola and rapeseed meal that we can use in animal diets because they contain high concentration of crude protein and amino acid and low level of glucosinolates. For variety and location of canola and rapeseed, there are two different varieties of canola and rapeseed that they grow in different regions around the world. Brassica napas that they can grow in Australia, Canada, China, Europe, and US. Brassica campestris or rapa that they can grow in Northern Europe, Northwestern China, and India. For the composition of canola and rapeseed themselves contain fat range from 42 to 43%, and crude protein range from 20 to 30%, depend on varieties of canola and rapeseed and growing condition that they grow in different regions around the world, as I mentioned before. And these differences could affect nutritional values of seed and consequently affect the nutritional values in meals. For oil extraction procedures, there are two different types of oil extraction procedure that they use to remove the oil from canola and rapeseed. They have expeller extraction and solvent extractions. For expeller extraction, they will use mechanical press to remove the oil from canola and rapeseed. The temperature for conventional expeller extraction could range from 95 to 130 degrees Celsius, depending on speed of screw that they use in the process. They also have another type of expeller extraction that they call cold press. They control the temperature in this type of process not greater than 60 degrees Celsius because virgin oil from this type of process still in demand of customers who prefer vegetable oil from the process without high temperature and chemical reagent. For solvent extraction, they will use both mechanical press and solvent to remove oil from canola and rapeseed. They will cook canola and rapeseed in cooking step before moving them to mechanical press to remove 60 to 70% of oil. Then the rest of the oil will be removed again in solvent extraction step that normally they use hexane. After that, the meal will be removed to the solventizer and toaster to recycle hexane back to use in solvent extraction step again. For chemical composition of canola and rapeseed, the meal from expeller procedure contain crude protein range from 33 to 35% and contain fat range from 9 to 13%. And the meal from solvent extractions contain crude protein range from 35 to 38% and contain fat range from 2 to 5%. By different oil extraction procedures, the chemical composition in canola and rapeseed could be different because the differences in oil extraction efficiency and heat exposures between expeller procedure and solvent extraction procedure could affect energy and amino acid concentration in the meal and consequently affect nutritional values of the meal. So now we know that two factors that could affect nutritional values of canola and rapeseed meal are locations that they grow canola and rapeseed and processing procedures that they use to remove oil from canola and rapeseed. Therefore, the objectives of this experiment were determining and comparing the digestibility of crude protein and amino acid in canola meal, 0 rapeseed meal, and 0 rapeseed expellers from different locations and from different processing procedures. For material and methods, we had 22 samples of canola and rapeseed meal. 7 samples were canola meal, 10 samples were 0 rapeseed meal, and 5 samples were 0 rapeseed expellers. We wanted to compare crude protein and amino acid digestibility in canola and rapeseed meal from different locations. So we compare canola meal from North America and 0 rapeseed meal from Europe. And we wanted to compare crude protein and amino acid digestibility in rapeseed products from different processing procedures. So we compare 0 rapeseed meal from solvent extraction with 0 rapeseed expellers from expellers procedures. We used 23 pigs at the initial body weight of 28.8 kilograms. Pigs were fitted with T canola that allow us to collect ileal digester to analyze and calculate the digestibility of crude protein and amino acid. We formulated 23 diets. 7 diets were formulated from each of 7 sources of canola meal. 10 diets were formulated from each of 10 sources of 0 rapeseed meal. 5 diets were formulated from each of 5 sources of 0 rapeseed expellers, and 1 diet were nitrogen-free diet to determine the basal endogenous loss of crude protein and amino acid. All 23 diets were formulated to contain the same concentration of crude protein and fat. Pigs were fed at 3 times the estimated energy requirement for maintenance. They were allotted in 23/9 Uden square design with 23 diets and 9 periods. 5 days for adaptation to the diet, followed by 2 days for ileal digester collection. Data was analyzed using prog mix of SAS. Diet was included in the model as fixed effect. Peak and period were included in the model as random effects, and we used contrast option to determine the difference of means at the alpha level of 0.05. Now, let's move into the results. We'll talk about the concentration of crude protein and amino acid in canola and rapeseed meal from different locations first. As you can see here in this bar graph, orange bar represent canola meal from North America, and blue bar represent 0 rapeseed meal from Europe. The concentration of crude protein in canola meal was 37.5%. The concentration of crude protein in 0 rapeseed meal was 36.4%. And the concentration of crude protein in canola meal and 0 rapeseed meal from different locations were not different. Same thing for the concentration of amino acid. The concentration of lysine, methionine, threonine, and tryptophan were not different between canola meal and 0 rapeseed meal from different locations. For the concentration of crude protein in rapeseed products from different oil extraction procedures, here we have orange bar represent 0 rapeseed meal from solvent extraction, and blue bar represent 0 rapeseed expeller from expeller procedures. The concentration of crude protein in 0 rapeseed meal was 36.4%. The concentration of crude protein in 0 rapeseed expellers was 35.6%. And again, the concentration of crude protein in 0 rapeseed meal and 0 rapeseed expeller from different oil extraction procedures were not different. Same thing for the concentration of amino acid. The concentration of lysine, methionine, threonine, and tryptophan were not different between 0 rapeseed meal and 00 rapeseed expeller from different oil extraction procedures. Now, let's talk about standardized ileal digestibility or SID. This bar graph represents SID of crude protein and amino acid in canola and rapeseed meal from different locations. Here we have orange bar represent canola meal and blue bar represent 00 rapeseed meal. You can see here that SID of crude protein, lysine, methionine, threonine, and tryptophan were not different between canola meal and 00 rapeseed meal from different locations. Same thing for other amino acids. SID of arginine, histidine, isoleucine, leucine, phenylalanine, and valine were not different between canola meal and 00 rapeseed meal from different locations. Now, let's talk about SID of rapeseed products from different oil extraction procedures. Here we have orange bar represent 00 rapeseed meal and blue bar represent 00 rapeseed expellers. You can see here that SID of crude protein, lysine, and methionine in 00 rapeseed expeller were greater than that in 00 rapeseed meal, but SID of threonine and tryptophan did not differ. For other amino acids, SID of arginine, histidine, isoleucine, leucine, phenylalanine, and valine in 00 rapeseed expeller were greater than that in 00 rapeseed meal. With that result, we observed that nutrient composition and SID of crude protein and amino acid are in agreement with the values of canola meal and canola expellers in NRC 2012, and we observed that nutrient composition and SID of crude protein and amino acid are not different between canola meal and 00 rapeseed meal from different locations. This may be because both canola meal and 00 rapeseed meal came from same variety, which is Brassica napus, and they use the same oil extraction procedure, which is solvent extraction, to remove the oil from canola and rapeseed. We also observed that SID of crude protein and most indispensable amino acids are different between 00 rapeseed meal and 00 rapeseed expeller from different oil extraction procedures. This may be the result of the differences in efficiency of oil removal from seed, which may affect the concentration of gross energy, fat, crude protein, and amino acid, and consequently affect nutritional values in meals. And heat damage during the solventization and toasting step of solvent extraction may also affect the digestibility of amino acid in 00 rapeseed meal when compared to 00 rapeseed expellers. In conclusion, for standardized ileal digestibility of crude protein and amino acid in canola meal and 00 rapeseed meal from different locations, we conclude that canola meal and 00 rapeseed meal are not different. For standardized ileal digestibility of crude protein and amino acid in 00 rapeseed meal and 00 rapeseed expeller from different oil extraction procedures, we conclude that 00 rapeseed expeller is greater than that in 00 rapeseed meal except for threonine and tryptophan. With that, we would like to acknowledge ADM, AgriFirm, Bunge, Cargill, and CP Group for funding this research. Thank you.